Good afternoon, everybody. Welcome. This is our second edition of our World Trade Center Health Program research webinars. We are very excited that you joined us this afternoon. We have a lot of great information to share today. I'm going to turn it over to Dr. Travis Cabali, our Associate Director of Research here at the World Trade Center Health Program. He's going to start us off with some fantastic announcements, and then we'll go from there.
Thank you very much, Rhonda, and welcome everybody. Just a couple of quick announcements. Rhonda and Ann will be sending around a recent publication related to Charlie's presentation last month. It's a cancer incidence study involving rescue and recovery workers, and that will be coming to you, like I said, in the chat. In case you missed the announcement this week, we wanted to also let everybody know as a reminder that this last week there was a forecasting posting indicating our plans for a new funding opportunity, R21 Exploratory Developmental Research call for FY23. It'll be one of three postings for FY23. And this one is soliciting grants that examine modifiable risk factors associated with chronic diseases. The last item I wanted to just point out before I turn it back to Dr. Anderson is that we're delighted to have Dr. Branch presenting today. As a reminder, the scope of the World Trade Center Research Program includes covered and also non-covered or emerging conditions. And this is one of several examples in the series of presentations that will cover these type of emerging condition topics. And with that, I wanted to turn this over to Dr. Anderson, who you all, I'm sure know. If you don't, she leads our clinical consultation team and plays a primary role in making sure that our research program and scope is integrated with clinical practice. And we're delighted to have her introduce Dr. Branch. Christy?
Thank you so much, Travis. And yes, I want to extend a warm welcome to everyone. I'm so glad you're here. I'm hoping you're excited as I am about hearing about this presentation. And I want to build on what Travis just mentioned about emerging conditions. The program is concurrently always assessing the clinical landscape. We want to be ready to translate the findings of the research program into our clinical operations and our healthcare benefit plan when the research aligns with the legislative authority. And I think it's an exciting time because there are several new drugs under investigation for fatty liver disease, where previously there have been none. One recently reported the first part, well, a pharmaceutical company recently reported the first part of their phase 3 clinical trials, which demonstrated safety for individuals using the medication chronically. And while lifestyle interventions are still an incredibly important management strategy for fatty liver disease, we can also see the possibility of an effective pharmaceutical in the future for select patients in this country. With that, I want to turn it over to Dr. Branch. Dr. Branch, Andrea Branch, is a professor of medicine and the director of the Basic and Translational Research in Mount Sinai Institute of Liver Medicine. She has extensive experience in environmental health, previously demonstrating proximity to Superfund toxic waste sites is a risk factor for cholangitis. Her current World Trade Center related research is focused on determining whether World Trade Center responders have an increased risk of liver disease as a consequence of their exposure, and to characterize the burden of liver disease in our population. Dr. Branch, the floor is yours.
Thank you. Let's see. Okay, I have a label that is preventing me from seeing what's going on. Can people see my slides?
Perfect. We can see it, Dr. Branch. Thank you.
Okay. So thanks so much for the invitation to present today, and thank everyone who's here to listen. Thanks to the program. It's just been a wonderful opportunity to investigate liver disease in people with exposures to the World Trade Center dust cloud. And it's been fascinating and very rewarding. The title of my project is Hepatotoxic Exposures, Progressive Fatty Liver Disease and Liver Cancer Risk in the World Trade Center Health Program General Responder Cohort. And down a little bit lower, I've presented a diagram that represents thinking about how toxic exposures result in liver injury and disruption of lipid metabolism involving both the liver and adipose tissue. And the concept that particularly adipose tissue can become a storage site for toxic materials. So there's an acute insult that both the fat tissue and the liver have to deal with, but then subsequently these stored toxins can be released over time and can give rise to chronic injury that can manifest itself in various organs. So here are my disclosures. And this is my research team that I've had really great pleasure to work with on this project. As Christy was mentioning, this project in particular combines health and research objectives. One of the co-investigators is Michael Crane, who is the director of the World Trade Center Healthcare Program here at Mount Sinai. And another key collaborator is Claudia Henschke, who is a pulmonary radiologist who's been instrumental in disseminating lung cancer screening really globally. And she's been doing really brilliant work developing new computer algorithms to extract information from chest CT scans. The team includes a number of hepatologists and computer scientists, statisticians, data analysts. This is Mark Miller, my research coordinator. So this is a list of our publications, and since the slides are going to be available, they'll obviously be recorded for everyone. This slide goes through the study's overview and its potential impact. So the aims were to develop and then to use innovative digital technologies to conduct the first systematic investigation of liver disease in World Trade Center responders. Our rationale was that the liver is one of the body's main detoxification sites, and numerous environmental and occupational toxins are known to cause liver damage. This damage can persist for decades after the exposure has ended. Our central hypothesis is that exposure to particulates and toxins in the World Trade Center dust cloud cause liver damage and increase the risk of end-stage liver disease and liver cancer. And our impact, we hope, will be that by uncovering previously unrecognized liver disease and by introducing new digital technologies, we will improve the health of World Trade Center responders and advance computational methods for analyzing CT images and medical record data. And we also hope to inform public policy about the health consequences of exposure to liver toxins. The three aims are to examine the relationship between the intensity of exposure to the World Trade Center dust cloud and the prevalence of liver steatosis, to develop computer algorithms to identify World Trade Center General Responder Cohort members who have liver steatosis, cancer, and other liver diseases, and to determine the prevalence of advanced liver fibrosis in the World Trade Center General Responder Cohort and identify risk factors for advanced fibrosis. This slide contains a small, tiny portion of the information that's been put forward about the contents of the dust cloud. And I just wanted to particularly highlight lead and PCBs because I'm going to speak about them a bit in particular. So one of the reasons why the liver is so often damaged by toxic exposures compared to other internal organs is because it plays a central role in detoxification. So it's really designed to take up toxic materials and particulates and process them for excretion so that they can be eliminated from the body. So there are many different steps in the process and many alternative pathways. This slide highlights the potential of environmental toxins to enter into the liver, to be acted upon and to activate the cytochrome P450 system in what is called phase 1 of detoxification, which frequently leads to activation of intermediates. It can also lead to deactivation of intermediates. But in any case, phase 1 prepares these incoming molecules for further processing by sulfation or conjugation, so that they're in a form that allows them to be excreted. That's on the one hand. And on the other hand, the liver is also the main site in the body for dealing with bacterial products that are imported across the intestinal wall and come up to the liver through the portal vein. And these materials are taken up primarily by the liver macrophage, the Kupffer cell, and degraded and turned over, so they don't make it into the general circulation. There's so many different pathways that I could point to as pathways that lead to disruption of liver function and liver lipid metabolism. This particular slide illustrates one pathway in the particular case of PCPs coming into the liver. The liver has a number of proteins that can bind ligands and become transcription factors. I've highlighted two different members of this family, the aryl hydrocarbon receptor and the constitutive androstenedione receptor. These have rather promiscuous binding sites, so they bind any of a number of molecules, including PCBs. And upon ligand binding, they enter into the nucleus and bind to their sites in combination with other parts of the transcription machinery and regulate the expression of downstream genes. Some of these genes include the P450 enzymes. And as a result of the activity of these enzymes and the generation of various molecular species, there can be disruption of mitochondrial function. And since the mitochondria is the main site of beta oxidation, in addition to peroxisomes, disruption of mitochondria can lead to lipid abnormalities. And this leads to the excessive accumulation of fat in the liver. There's a long list of publications that document the hepatotoxic nature of many workplace and environmental toxins. And I just particularly want to highlight the top study that I've listed here by Matt Cave, who really coined the term toxicant-associated steatohepatitis, which is a severe form of liver injury, which he characterized in workers in vinyl chloride plants. So many exposures can damage the liver. These can be toxicants, alcohol, materials from our diet, getting into the liver. And down at the bottom, I've illustrated the changes in the liver that can occur. So here on the left, you see steatosis. All these white globs are fat globs in the liver. And this can proceed to steatohepatitis, which is a combination of fat in the liver with inflammation, damage to hepatocytes, and the blue stain material is the beginning of scar or fibrosis. This can progress to cirrhosis, where you have separate nodules of the liver that are walled off from each other. And liver cirrhosis is a very important precursor condition for the development of hepatocellular carcinoma, which is the most important of the different types of primary liver cancer. It's about 80% of all primary liver cancer. So the features of toxicant-associated fatty liver disease and its severe and progressive form of toxicant-associated steatohepatitis are the following. And it's important to note that liver toxicity is the most common organ injury due to occupational environmental exposures to industrial chemicals. A wide range of liver pathologies, including cirrhosis to liver cancer, can follow chemical exposures in both humans, and this has been well characterized in a number of animal models. TAFL-D and alcohol-related liver disease, and another sort of grab bag of liver disease called non-alcoholic fatty liver disease, or NAFLD, can all look quite similar to each other in histopathology. As I mentioned, TASH is the severe form of TAFL-D, and it's characterized by steatosis, inflammation, and fibrosis. Subjects with TASH, by definition, have exposures to chemicals such as vinyl chloride. They may also have traditional risk factors for liver disease. Obviously, being exposed to liver toxicants in your workplace or environment doesn't preclude also having liver damage from additional sources, such as viral hepatitis, alcohol consumption, and obesity. And in fact, all these exposures often act in an additive or synergistic manner to cause liver disease progression and end-stage liver disease. This is a bit of local color. That Mount Sinai was one of the first institutions to recognize the existence of TASH. It was reported in the New England Journal of Medicine, and Irving Selikoff was a co-author of this study. And is noted in the bottom, from the very beginning, it was recognized that one of the frightening aspects of exposures to these liver toxicants was that the injury persisted for decades after the exposure had ended. A few features of TASH. In the A panel up at the top left, you see really the characteristic feature, those white globules of liver fat. In Matt Gave's study of 25 workers with vinyl chloride, he found that combination of fat inflammation and hepatocyte damage in 80% of the workers who had liver biopsies. About 30% had fibrosis. He found insulin resistance, but very little diabetes, and I'll return to the diabetes later. He showed progression. A number died of liver cancer or cirrhosis, and other deaths included pulmonary disease and congestive heart failure. And I will mention these later in my talk as well. So the study group for this project is the general responder cohort. This slide lists the eligibility criteria for being a member of this cohort, and also the data that are collected and the intervals of data collection. And this really forms the basis of the information that we had to investigate in this study, noting down at the bottom that many members of the general responder cohort also received healthcare at Mount Sinai outside the World Trade Center. And we have IRB approval to examine those medical records as well. So that really increases the amount of information that we can collect and analyze. The types of liver-related data that we have access to are summarized here. Liver fat, fatty liver, or liver steatosis, as it's called, can't be identified by any blood test. And that is definitely a problem. However, many members of our cohort have had chest CT scans, often because they had respiratory problems and had chest CT scans to investigate the respiratory problem. And there are methods available, and I'm going to talk to our work developing very facile one that can take chest CT images and determine whether a patient does or does not have liver steatosis. we can estimate the stage of liver fibrosis based on age, AST, ALT, and platelet counts. And in our analysis, we use a cut point of a FIB4 score over 2.67. We selected this cut point because in previous studies of the NHANES cohort, this cut point was associated with the 25-fold excess of liver-related mortality. So we feel that it is a threshold that identifies a high-risk population. We also have information about the audit score, anthropomorphic data that lets us assess BMI, body mass index, data about comorbid conditions that are self-reported. And for liver cancer, we've really used a combination approach of developing software that we can use to identify individuals with primary liver cancer from their medical record, which we then verify by chart review, but also from data from the World Trade Center Data Collection Center and from Dr. Emanuele Tuoli, who's the director of the World Trade Center Specimen Bank. So I think some people might find it surprising that we use chest CT scans to identify liver steatosis, but as this diagram illustrates, these chest CT scans actually get a very large portion of the liver, and that provides us the material that we need to analyze in order to identify patients with steatosis. When the liver accumulates excessive fat, there are really changes that can be observed at all levels. So this top part of the slide illustrates the normal liver, both at the level of a liver biopsy or histopathology, in the center, general liver anatomy, and there on the right is the CT scan, and the liver is marked high Hounsfield units, and that's because a normal liver attenuates the CT signal to a significant extent. So in contrast, in a patient with steatosis, a liver biopsy shows large globules of fat. The liver itself, if you have gross anatomy, shows a lightened color and is often enlarged. And in a CT scan, there is low attenuation. So the liver appears darker when there is fat in the liver because there is less attenuation of the signal. So this change in attenuation of the CT signal that occurs as the liver fat content can be turned into a quantitative assay. In our initial investigation of liver steatosis, we used manual methods in which the liver was first divided into different quadrants. A region of interest in each quadrant was identified by a radiologist. The attenuation in Hounsfield units was then measured and summed and averaged. And this slide is just to let everyone know that all steatosis is not identical. When it's analyzed by biopsy, it is scored as mild to moderate and severe. Chest CT scans are not a particularly sensitive way to pick up liver steatosis. So mild steatosis, we would not be picking up. So what we're picking up when we call a patient is having liver steatosis and it's determined by CT, we're talking about moderate to severe steatosis. And what that means is that if you did a histological examination, you would find a minimum of 30% of the hepatocytes have these large fat globules in them. One of the studies that we did as part of this project, and again, I really am very grateful to the World Trade Center Health Project for funding our work because I think it's been important and very rewarding, was to do an analysis in which we compared liver steatosis in members of the general responder cohort to a control population. In this study, all subjects gave informed written consent, and all members of the World Trade Center general responder cohort qualified for lung cancer screening. Liver attenuation was measured on low-dose chest CT scans. Moderate to severe liver steatosis was defined as attenuation below 40 Hounsfeld units, which is an established measure. Bivariate and multivariable linear and logistic regression were performed. Propensity scores were calculated, and inverse probability weighting was used to adjust for potential variables in a comparison of World Trade Center to non-World Trade Center participants. So before comparing the World Trade Center and the non-World Trade Center group, we first compared people in the World Trade Center cohort who did and who did not have liver steatosis. And one of our important findings was that the group on the left with a Hounsfeld unit less than 40 had a significantly higher percentage with elevations of what we call liver enzymes, aspartate aminotransferase and alanine aminotransferase, ALT. So down at the bottom, I've highlighted that the responders who had liver steatosis, 40% of them had an elevation of liver enzymes, and this compared to only 6% of the cohort who did not have liver steatosis. So there's a high likelihood that when we identify responders who have liver steatosis, they don't have what could be called bland steatosis, which is simply excess fat in the liver. They have fat in the liver and they have liver injury as well. And that indicates a individual who is at risk for progressive liver disease. Interestingly, on further analysis of factors that were associated with having liver steatosis among members of the general responder cohort, in logistic regression, the only factor we identified that was associated with liver steatosis was the coronary artery calcification score, which can also be calculated from chest CT scans. So we are not currently, but are very interested soon in exploring the relationship in the World Trade Center cohort between liver steatosis and other abnormalities such as coronary artery calcification. Here we did compare the attenuation values of the World Trade Center and the non-World Trade Center groups. And this slide is showing that the attenuation signal was lower in the World Trade Center cohort, which are the blue bars, than in the non-World Trade Center cohort. I'm sorry, World Trade Center in the pink, non-World Trade Center in blue. And the fact that the curve is shifted to the left indicates that members of the World Trade Center cohort had more steatosis. On analysis at the bottom of the slide, you can see that the mean value, the mean attenuation value was lower in members of the World Trade Center cohort, and a higher percentage had liver steatosis, 16.2% versus 5% in the non-World Trade Center cohort. We then used propensity and inverse probability weighting to assess whether membership in the World Trade Center cohort was an independent risk factor for liver steatosis. In this analysis, the propensity score was calculated for each participant using logistic regression analysis. The explanatory values included all measured covariates that were available for both groups, and they're listed here. Inverse probability weighting was then performed as outlined here. And what we found in this analysis was that membership in the World Trade Center cohort was associated with a 3.4-fold higher risk of steatosis after adjusting for a number of potential factors. So this investigation demonstrated that liver steatosis was associated with liver injury in World Trade Center responders, as indicated by higher measurements of AST and ALT. Steatosis was associated with higher coronary artery calcification scores in a multivariable logistic regression model. Exposure to the dust cloud was independently associated with a 3.4 higher odds ratio for moderate to severe liver steatosis. And these findings really are consistent with two other investigations that have been reported on individuals with exposures to the World Trade Center dust cloud. The first report by David Prasant and Michael Wyden, who found 22% of the firefighters in their cohort had elevated steatosis, and a group from New Jersey who reported at the DDW meeting in 2020 that 83% of their group had fatty liver. But I've been unable to find a publication of their study, so I did not include the details here. But if I do get a copy of their abstract, I'll include it in the online version. So having completed that study, we're very enthusiastic about going forward with additional investigation of steatosis and needed a way to accelerate the process. To do this, we developed a computer algorithm to automatically identify patients with liver steatosis. And our approach is published in the European Journal of Radiology. The algorithm is based on the use of a number of established anatomical landmarks, such as the top of the right lung and the top of the diaphragm, from which the program then projects 70 millimeters down and enters into the liver. The method for determining the attenuation value, and down below I'm showing the lungs in pink, overlying the liver in red. So first, the lung is segmented using published methods. The top of the diaphragm under the right lung is identified. A projection is made down from the lung, 20 millimeters, into the liver, which is then divided into regions of 1 cubic centimeter. Regions that are outside the interquartile range of attenuation values are excluded, and then the mean of the remaining cubes are averaged to determine the attenuation value in Hounsfield units. This slide shows a comparison on the y-axis of the measurements made by the algorithm, and on the x-axis measurements of the same CT scan that were determined by manual measurement. The R-square is 0.99. So we conclude that the algorithm provides an automated, robust, and accurate method for identifying individuals with moderate to severe liver steatosis based on non-contrast chest CT images. So we then went on to use the algorithm to analyze 1,788 chest CT scans of members of the World Trade Center General Responder Cohort. In this analysis, we used the earliest available low-dose chest CT scan performed on members of the General Responder Cohort from 2003 to 2008 as part of the World Trade Center Health Program, who also had laboratory test data available for analysis within 12 months of the CT scan. Dust cloud exposure was categorized based on World Trade Center arrival time. Liver attenuation was determined using our computer algorithm, and the dose-response relationship between intensity of exposure to the dust cloud and the risk of moderate to severe liver steatosis was evaluated using linear regression and other methods. General linear models were used to compare the mean liver attenuation between exposure groups, adjusting for sex, age on September 11th, race, smoking status, BMI, alcohol use, diabetes, GERD, and the forced expiratory volume in 1 second, or FEV1. And what we found is illustrated on this slide, and it is that individuals who had an earlier arrival at the dust cloud had an increased prevalence of liver steatosis that was statistically significant. So our findings are important because they have produced a computer algorithm that is available to automatically identify individuals with moderate to severe liver steatosis. And of course, this could be applied to any chest CT scan, non-contrast chest CT scans. This is an important clinical advance because liver steatosis is often associated with progressive liver disease, and liver steatosis cannot be detected by any blood test. The dose-response relationship between the prevalence of liver steatosis and the intensity of exposure to the dust cloud increases prior evidence that toxicants in the cloud cause significant and persistent liver damage. So I now want to switch to talk about our third aim, which was to determine the prevalence of advanced fibrosis in the World Trade Center general responder cohort and to identify risk factors associated with fibrosis, which is scarring, as opposed to steatosis, which is fat, as I talked about in the previous slides. This is a reminder that there are cellular and molecular responses to liver injury. The liver injury can come from different directions: viral hepatitis, alcohol, metabolic syndrome, toxins. Can lead to fibrogenesis, scarring, cirrhosis, which has a consequence of portal hypertension and bleeding. Scar is the histological feature that is most strongly associated with liver disease progression and mortality. So it's particularly important to identify fibrosis so that patients can be referred for care. And of course, in addition, you also have steatosis, inflammation leading to steatohepatitis, and a reminder that what this all leads to is damage to liver cells. And of course, since the liver is a vital organ, the loss of liver function itself is a health problem. Here you see, once again, these white globs just to remind you what we're talking about. This is fat in the liver. These small purplish cells are inflammation in the liver. And this blue stain material is extracellular matrix, scar, fibrosis. Much of it is collagen in the liver. And what this slide shows is the relationship between the increase in the stage of liver fibrosis, and here all-cause mortality, and here liver-related mortality. And these bottom two slides show the mortality rate ratio. So you see there's quite a steep increase in liver-related mortality as you have an increase in the fibrosis stage. So in order to examine fibrosis in the World Trade Center cohort, we really wanted to establish a foundation using some of the tools we would be using, applying these tools to a well-characterized cohort of patients, and we turn to information available from the National Health, Nutrition, and Examination Survey and Haynes. So this next section is very much work in progress. We combined 10 cycles of the NHANES and then selected individuals for whom the FIB-4 score and the FORNS score, which is a related index that allows fibrosis stage to be identified, and excluded people lacking data, pregnant women, individuals less than 20 years old, and we ended up with 47,000 members of the NHANES cohort with data we could analyze. And what I've blocked off here is our analysis by race of the prevalence of what we call advanced fibrosis. So by advanced fibrosis, I mean stage F3, F3 and F4. So these are people with an elevated risk of liver-related mortality and an elevated risk of all-cause mortality. And what we found was that looking at this national population provided by NHANES, the race and ethnicity group with the highest prevalence is African Americans, non-Hispanic Blacks. We then went forward looking at the relationship between advanced liver fibrosis and risk factors that are commonly associated with liver fibrosis in the general population, but this time stratifying by race. And what we found was that diabetes, which is very commonly noted as an important fibrosis risk factor, actually did not appear to be a risk factor for non-Hispanic Blacks. And it did not appear to be a factor for non-Hispanic Blacks, regardless of how we defined diabetes. Whether we define diabetes as self-reported diabetes by hemoglobin A1C or by fasting glucose, even though we found a strong association between diabetes in non-Hispanic whites, Mexican Americans, and the other group, we did not see this association in African Americans. We conducted a multivariable logistic regression and were able to show in this that, in fact, diabetes was not associated with liver fibrosis in non-Hispanic Black persons, although it was associated with fibrosis in other groups. On further examination, we were surprised to find that, contrary to our expectation and contrary to example for what is seen in Mexican Americans, where you have a strong increase in the prevalence of advanced fibrosis as you have an increase in BMI or an increase in your waist circumference, in African Americans, we actually found an inverse relationship with the highest prevalence of fibrosis in Black persons of normal weight and normal adiposity as indicated by a normal waist circumference. So we then sought additional risk factors that might be drivers of fibrosis in this cohort and investigated in this slide lead. And what we found was that non-Hispanic Black persons actually had the highest prevalence of lead in the fourth quartile of lead blood levels, which is particularly unfortunate because when we then looked to see if there was a relationship between exposure to lead and fibrosis, we found that African Americans in the fourth quartile of lead levels had a significant increase in the prevalence of advanced fibrosis. And when we did a further stratification of individuals in the fourth quartile of lead exposure, we found that among African Americans, being of a normal body weight was associated with a significant increase in the prevalence of advanced fibrosis. So liver fibrosis was most prevalent among non-Hispanic Black persons with high lead exposure and normal body weight. We then explored other toxicants to see if we found an association between them and liver fibrosis. And indeed, we found that high PCB exposure is strongly associated with liver fibrosis in the NHANES population. So We then did an investigation where we matched members of the general responder cohort to members of the NHANES cohort. These are very, very different cohorts. And in order to do a one-to-one matching, we ended up with 7,493 in each group. When we did a logistic regression analysis on this subset of the general responder cohort, we found risk factors for advanced fibrosis included better pulmonary function. It did not include diabetes. And we found that there was a trend toward a higher prevalence in non-Hispanic Black persons. In contrast, when we did a multivariable logistic regression on the NHANES cohort that we had selected in our one-to-one matching, we found that better pulmonary function was not associated it was not associated with liver fibrosis in this population. So our interim conclusions and future directions are: the NHANES data suggests that exposure to toxins, and we particularly looked at lead and PCBs, may be important contributors to liver fibrosis in the United States. Non-Hispanic Black persons, especially those of normal weight, may be particularly at risk for liver disease caused by toxic exposures, and the risk factors for advanced liver fibrosis may differ between members of the general US population as a whole and the World Trade Center general responder cohort. The World Trade Center specific risk factors may include Black race, ethnicity, and pulmonary dysfunction. So I just want to close with some final information about additional computer algorithms that we developed as part of this project. These algorithms we developed in order to analyze medical record data. We have published 2 sets of algorithms, and together they allow us to identify individuals with fatty liver, liver cancer, and viral hepatitis. And I would say that a key feature of our algorithms was our use of natural language processing to find reports of steatosis and liver cancer in radiology reports and pathology reports. And the HCV case finding algorithm, which was published a few months ago in Hepatology, is available on GitHub. It's really, really important to do surveillance for liver cancer because it's really the only way to detect liver cancer when it's at an early stage. These 2 slides compare survival in people who are less than 70 years of age. The colored lines indicate different time periods, with the most recent time period being shown here in brown. When liver cancer is detected before it has metastasized, it still has very poor survival of less than 2 years. But when it is detected only after it has metastasized, it has a median survival of about 6 months. So we really want to be as vigilant and helpful as we can be to identify patients who are at risk for progressing to liver cancer and engage them in surveillance programs. So far, we have done an analysis of 30 members of the general responder cohort that we had identified using our computer algorithm by 2018. And this slide shows a description of these 30 cases. But we recently have received additional data from the Data Collection Center, and we now have 40 newly identified cases to analyze. So we will certainly be working on that and trying to get to the bottom of the cause of their liver disease, which we expect to be a combination of viral hepatitis, alcohol-related injury, metabolic syndrome, and TASH. So this slide is just a final reminder that you really need to go out and hunt for liver disease in an asymptomatic stage. because liver disease causes few, if any, distinctive symptoms until late stage disease. So it's the opposite of a situation where somebody loses a front tooth. Those people are going to go to the dentist, whereas a person with a grave liver condition may not come to the attention of any hepatologist until there's very little that can be done. So we want to raise awareness and spread information about the need for vigilance and referral to hepatologists. So the conclusions of our project are that World Trade Center exposure may increase the risk of toxicant-associated steatohepatitis, liver fibrosis, and liver cancer. Algorithms to automatically detect moderate to severe liver steatosis in chest CT scans and to identify fatty liver disease, HCV, and liver cancer based on EMR data were developed and applied. And we're working to identify responders at high risk of liver disease and liver cancer and help them access liver care and healthy lifestyle interventions. And as Christy mentioned, we are on the cusp of having novel treatments for fatty liver disease. And certainly here at Mount Sinai, we have many clinical trials of investigational drugs and are eager to enroll individuals in these trials. And in addition, there are wonderful drugs that are available for the treatment of viral hepatitis. And hepatitis C can be cured in 12 weeks, one pill once a day for 12 weeks. So we want to make sure that everyone who could benefit from these interventions has that opportunity. And finally, additional research is needed to define the relationship between exposure to the dust cloud and liver disease. And I've got so many people to thank, most especially the responders. Big thanks. Andy, Todd, Christopher Tesaro, and the whole data collection team, so many thanks for their help. I've mentioned Michael Crane. He's there with Claudia Henschke. This is Doug Dietrich, who is the head of the Institute of Liver Medicine here at Mount Sinai. Allison Hardy is a nurse practitioner who works with Doug. Amreen Dinani is a hepatologist on the project. Pani Perumaswamy and Mina Benzal are the other hepatologists. This is Artit Jirapatnikal, who is a computer scientist who developed the algorithm for CT analysis. This is Frank Ng, who has been working on my lab and some lab-based investigations relative to this project. Lee is a medical informaticist. Brooke Wyatt is a data scientist and statistician. Mark Miller is my research coordinator. Rowena Yip is Claudia Henschke's truly fantastic and talented statistician who helped in the design of the studies that we executed, and worked in collaboration with my PhD student, Ning Ma, to carry out the actual analysis. This is Sarah Lewis, the abdominal radiologist on the project who worked with us to validate the findings of the chest CT scan by analyzing abdominal MRI data. And this is Max Van Drome, who's the computer scientist in the group who wrote the algorithm to extract data from electronic health records. And of course, I want to thank NIOSH for supporting the project. And I also want to thank Charles Hall for starting off the series, getting the series off to such a great start last month. So thanks to everybody. And if there are questions, I'm available.
All right. Well, everyone have a fantastic day. We will be in touch via email, and we'll also let you know later on in the future where we can house these amazing recordings. You know, we kind of have to go through some processes here on our governmental side to make sure we can do that. Many thanks. Have a beautiful evening, beautiful rest of your day. Thanks again, Dr. Branch, and have a fantastic rest of your Thursday.
Thank you. Bye-bye.
Bye, everyone.